Hello, my name is Tyler Marmot.
And I'm Cody O'Connell. Welcome to the Biology Bakery. On this episode of Developmental Desserts, we're going to be whipping up some sweet acidians today, aka
Sea squirts.
So, Cody, some of our viewers may be a little confused as how we get from one cell to a complex organism like our acidian here.
How do they do that?
Well, I guess it all starts with maternal determinants.
Hi, I'm Alexa Feist, and I'm the sous chef here at the Biology Bakery. I'm here in the kitchen early today making maternal determinants, just as a mature acidian would before fertilization. Maternal determinants are mRNAs and proteins that are set up in the embryo by the adult acidian in specific locations. This is essential for cell differentiation and development. The simplified version of the central dogma of molecular biology is the process of DNA being transcribed into RNA, which is then translated into proteins. For example, the adult acidian transcribes MACHA1, an mRNA, which the embryo later translates into muscle cells. Another example is the maternal determinant protein beta-catenin, which signals particular embryonic cells to become endoderm cells at the 32 cell stage. For example, this maternal determinant is called MACHA1. It's localized in the PVC region of the embryo, and later on it'll encode for muscle cells. Another example is beta-catenin. This is a protein that when transported to the nucleus, it activates transcription factors that then signal the cell to form differentiated endoderm. There are many important maternal determinants that we don't have time to go over, but I will be preparing them in the bakery to put them in the correct place in the embryo so cell differentiation and development proceed smoothly. So all of these maternal determinants are placed correctly, and this is a nice looking embryo. So now I'm going to place it in the freezer to get it ready for cell division tomorrow.
Hey, let's check and see if Alexa separated the maternal determinants correctly in this embryo. What are you doing?
All right.
Let's
and,
do it.
You can see if it was fertilized as well.
Let's see where that nice embryo is. Ooh.
Oh my.
You can see a nice localization of that PVC. What is the PVC?
PVC stands for the
Posterior vegetal cytoplasm.
So Tyler, how do you know what the axes are of this acidian embryo just based on the maternal determinants that Alexa provided for us?
Well, if there was no PVC, it would be very tough indeed to having only blue and yellow on the top and bottom.
Almost impossible.
Impossible indeed. Yet, with the PVC, its name even implies where it localizes in the cell.
Posterior.
Excellent. And ventral on the bottom, as we can see here. Note it is not on the front half, only on the back. Because of this, we know that this side with the red PVC is the posterior half, whereas this half is the ventral half. And in acidians, there are different colors normally. So this red section is the PVC, which is clearly localized itself in one side.
That's going to play a key role in the cell divisions, which we're about to get started right now.
Let's do it. In order to divide the cell properly, we need our handy cell divider. Now this piece of twine will represent...
Really high tech.
Oh, it's incredibly high tech. This piece of twine will represent the invagination of the cell membrane during division. Now, all naturally occurring acidian embryos will divide the same way, starting with determining the left and right axes. And if you look here, you'll notice that the maternal determinants will be divided evenly, like so.
Voila! First cell division.
As you can see, the PVC and all the other maternal determinants are evenly distributed between the two halves. making the left and right halves of the embryo. Now, where do you think the second division will be? What axes will be made?
Anterior, posterior.
Excellent. That's correct. Anterior and posterior. Now, that would mean that our divisions would have to take place and look something like so.
Voila,
Excellent.
division number 2.
Now, if we break this apart only along the second division axis, we can see that the front half has no PVC at all, whereas the back, posterior half, has all of it. This will be very important later on in development.
Where's the last cell division then?
The last cell division before the 8-cell stage happens to create the dorsal and ventral axis. Now this one will be a little more tricky to
A
do.
little tricky, but
But it
the
should
cell
be
knows how to handle it.
just fine.
Boom.
Perfect.
Semi-perfect.
Here we have our 8-cell embryo.
And as you can see, all these blue ones on the top of these top 4 of the 8-cell stage are all going to be the ectoderm. And all these other guys will turn into differently specified cells. Later we will talk about. So Tyler, how is it that only Macho 1, the maternal determinant set up by Alexa, dictates the cell differentiation of our embryo here?
Well, maternal determinants aren't the only factors that help the cells become what they will eventually become.
What else is?
There's also cell-to-cell signaling. A key cell-to-cell signal is the FGF pathway. Something that Alexa will go into more detail.
This is the FGF pathway that dictates the differentiation of mesenchyme and notochord cells at the 64-cell stage of development. The ligands that are given from the endoderm activate the transmembrane receptors that are set up as a dimer that phosphorylates RAS, which will activate the RAF-MEK pathway, translocating MEK to the nucleus. There, it activates transcription factors that activate genes coding for mesenchyme and notochord cells.
So looking at this a little more detailed, we can see more clearly that the PVC is localized only in these 2 posterior ventral cells.
That looks like a really nice embryo.
It is very pretty indeed.
Alright, and as you can see, we're at the 8-cell stage at this point. We have the 4 on top. We're going to divide into ectoderm cells. The red right here is going to be muscle cells. In between the muscle cells and the endoderm, which is the yellow, will be mesenchyme. On this side, we're going to have nerve cord and notochord cells.
And this correlates with the axes. The PVC, the posterior vegetal cytoplasm, is going to be on the posterior side. It's not expressed in the anterior side. So, and we ignored the dorsal cells, the ectoderm, and the ventral side is what we've been focusing on, leaving the right and left axes. At the 64-cell stage, that would mean the FGF signaling pathway would come up positive for both notochord and mesenchyme, but it would not go in the nerve cord and muscle cells.
And based on what we already know and discussed about the PVC, having localized the Macho-1 RNA, the maternal determinant that Alexa set up for us, we can see that it's only localized in the PVC region. So that's why you'd have a plus here containing Macho-1 and a negative here not containing Macho-1 in the anterior vegetal cytoplasm. So Tyler, a lot of our viewers ask us how you get different cell types from only 2 cell signalings interacting.
Well, if we refer to the chart, we can see that there are 4 different cell types created through 4 different combinations of plus signs and minus signs using only the 2 pathways. Looking at our little example here, in between the red and yellow will be where the mesenchyme forms because it gets both signals. And at the top of the yellow right here, on the other side, on the anterior side, this would be where the nerve and notochord are formed. At this time, the cells have become specified enough that we can put them in our time oven to become determined.
What does specified mean?
Well, specified means that there are still several cell fates that these cells can become. Whereas determined, there are no more choices. That cell is set in its ways. It's either a heart cell, a skin cell, or an eye cell.
A couple weeks later.
Alright, Tyler, we've been waiting long enough. Let's see how the sea squirt's doing in the time oven.
I'm ready.
I'm pretty sure it's ready.
I'm so ready.
Whoa! It's looking so solid. Check a look.
Time oven does it again.
Perfect. Every time. Alright, let's take a look at this real nice adult sea squirt we got coming on.
Good stuff.
It's looking really nice, sessile, stuck to the substrate. It will not let go. It's pretty much going to be stuck there for the rest of his life.
Well, that's completely different from its juvenile form, which is free swimming. It finds a spot and sets up shop like this.
Look at, you see the real prominent siphons that they use for filter feeding with the water in, water out cycle. Really, really nice structures there.
It's a beautiful adult. It can only be possible thanks to the maternal determinants prepared by Alexa and our impeccable science.
We did it again.
Genius.
Woo!
Yes!
Perfect Ascidian development every time. I love it.
And now you can use the Ascidian embryos you develop in your own home to slow down any potential pirates you may encounter, so you can plunder your own booty.
Tyler, I don't think any viewers at home have a pirate ship they need to slow down. But you are totally correct. You can develop your own Ascidian embryos in your own kitchen with maternal determinants you have laying around.
Tune in next time when we teach you how to make a viable velociraptor embryo out of any spare fossils you may have lying around.
That's going to be a great episode, so be sure not to miss out. For all of us here at the Biology Bakery, tune in next week. Thank you very much.
And keep on developing.